Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. In this week's episode of Careers in Discovery, Emma is with Lucas Arruda, CSO of Curacell, a biotech on a mission to push the boundaries of cancer treatment. Lucas talked to us about his path from academia to industry, his lifelong love of science, and his desire to make a real difference in people's lives. We talked about the unique challenges of his role and managing a small team in a high-stakes environment.
Welcome back to Careers in Discovery. I'm joined by Lucas Arruda, CSO from Curacell this morning. Welcome, Lucas.
Welcome. Thanks, Emma. It's a pleasure to be here.
So I suppose I'm really interested in firstly hearing about Curacell and the mission towards bringing a life free from cancer. Fascinating work you're doing.
Yeah, before I start talking about the company, just want to give a bit of context. We work with cancer, as you said, and cancer, it's a quite complicated disease with several millions people dying per year. And there is quite a lot of development of using cell therapies to treat cancer, especially on the CAR-T field, which it works quite well for blood malignancies. But most of the cancers are solid tumors where the CAR-Ts don't work as expected. Yeah. So we think that by using the natural response to treat cancer by using TILs, tumour-infiltrating lymphocytes, we could expand the application of cell therapies from not treating blood malignancies, but also treating solid tumors. Just to give a bit more background, TILs, or tumour-infiltrating lymphocytes, they are our naturally occurring immune cells that can recognize and attack and kill cancer cells. They do that because they express a quite powerful scaffold of molecules called T-cell receptors, which is insanely diverse and quite powerful and can dictate if a T-cell is going to respond and kill or modulate a microenvironment. And that's unique to each person. So when the cancers are evolving and metastasizing and they're creating new mutations, normally they can evade from these TILs. And these TILs, they are normally specific for cancer-specific mutations. And basically what we do, we just take them out, expand them into billions using our expansion protocol, and then give them back to the patients. Yeah. And that's the main goal of the company right now, to expand the application of tumour-infiltrating lymphocytes from hot tumors. So this therapy has been approved recently, early this year, by Iovance, that has now a new product in the market to treat melanoma, which is how we call it a hot tumor. It's because this type of cancer has a lot of mutations. Therefore, there's a lot of immune cells in the tumor tissue that can be expanded by using these protocols. But it's only a small fraction of all the tumor indications. So our idea is that by optimizing the expansion protocol and also by having a new way of clinical development, we can expand that into cold tumors. So cold tumors, they have less mutations, but we have some data from patient-named treatments where we can still we could show that we can still expand tumor-specific T cells from cold tumors, even if you don't have such a high number of mutations. So if you have an optimized expansion protocol, you can make that happen. And that's the goal of Curacell, is to expand the application of TILs from melanoma, which is a quite crowded indication right now. Everyone is developing new therapies for melanoma, and bring that into other indications such as prostate cancer and etc. Colorectal and other indications that are more in the cold space.
Yeah. So it sounds like the possibilities are endless.
Yeah, exactly. Yeah, that's
bar the finance aspect, of course.
Yeah.
So I noticed that you're started working on prostate and colorectal cancer. How do you select those indications? You talked a little bit about the crowded marketplace, and I suppose that's important criteria.
Yeah, that's a very good question. The idea of treating prostate cancer is because it's a quite prevalent disease. It's a large indication, and despite the survival rates are quite good if you are diagnosed early and you
Yeah.
have a stage one localized or
Mm,
stage two with more regional disease, it's quite aggressive and bad if you have stage
yeah.
four. So when the disease starts to spread into the distant organs, the mortality rate is as low as 32% in 5 years.
Wow. Yeah.
And because it's a quite prevalent disease, and with very few alternatives for this patient group that are refractory to radiation therapy, to surgery, or hormonal castration, we thought that it would be a good space to start with.
Wow.
And that also came from early observations in patient-named treatments. So during our preclinical development and learning how to expand the cells using a combination of cytokines, we had the chance to treat some patients in Germany using patient-named treatments, using some the company is based in Sweden, and here we could not run this clinical application because of the regulatory agencies. But in Germany, it's quite
Okay.
it's a bit loose. So you can have a patient exemption based on the agreement between the doctor and patient, and we
Right.
could treat one prostate cancer with our expression protocol and clinical application, and it worked quite impressively. So after
Brilliant.
infusions of our tool product, the patient achieved complete response. It's now
Oh my
more than five
gosh.
years free of disease. So
Wow.
then
That
that
enables you to collect the data you need.
exactly. So
Yeah.
along with the treatment of other patients as well, in all of them with cold tumors. Well, we also treated, for example, renal cell carcinoma patient that
Okay.
achieved quite a
Oh,
good disease, glioblastoma, pancreatic cancers. So all these patients, they had some of them complete responses, quite dramatic, some of them partial responses or stable disease, but overall indicating that we could apply this expansion protocol into a more cold space. And but
brilliant.
prostate, it's a must go because of the size of the population that unfortunately
Population.
is affected. Yeah.
Yeah. Okay. So, as a CSO at Curacell, how do you spend your days?
Yeah, that's also a good question. So we are quite a small company. Yeah, just a handful of employees based in Stockholm, and our manufacturing site is done in Germany by our CDMO. And basically, I supervise all the R&D activities. By being such a small company, I do everything from
Yeah.
bioinformatics, going to the lab if necessary. So, like a couple of weeks ago, I was making buffers. It's something that you normally don't hear from
Brilliant.
us. So I make buffers, run some experiments, drafting protocols, and most of the time just actually drafting protocols, discussing the results with the R&D team, and yeah, doing a lot of bioinformatics as well, and whatever comes, pitching
Yeah.
to investors, etc.
Yeah. In the lab one day, in the boardroom the next.
Yeah, I mean, we move as we have to. We just follow the flow, basically.
Yeah. Okay. I'm keen to take it back a step and talk about your early career and how you decided on a career in science, particularly in drug discovery.
Mhm. Yeah, since I was young, and it's funny to talk to my mom and to my family about where I
Mm,
am right now and how I was as a kid. I always wanted to be a scientist,
okay.
regardless of the field.
Did
My
you
dad
did you
was
have people in your, I was going to just ask, do you have scientists in the family to model that?
actually not. No, that's a good
Okay.
point. Actually not. I mean, my father was an engineer and my mom is a teacher. So at home we always had some cool stuff about. I mean, my mom, because she was a teacher, she always helped us with homework and making us, to follow everything and to learn everything the best out of
Mhm.
it from school. But I always wanted to be a scientist just for sake of helping people and learning new things and see how we can move the world and society and everything
Yeah.
forward. And I went to pharmacy school. Uh, yeah, because when I was learning about the professions and what I could do, I mean, I worked with my dad for a while as a junior engineer,
Mm,
with just to learn new things, and I did some courses on that area. And maybe I should have been pursuing that because that was quite fun. But I think that by going to medical sciences, you can help people in a different way. So I decided to go to pharmacy school because you learn a lot about drugs and you learn a lot about the body and how to develop new things. And you can apply that to thousands and thousands of people if you create a new drug. Of course, I mean, a medical doctor has that personalized treat and following up patients, but I never wanted to be a medical doctor. And then I decided to go to pharmacy school to get this good background from both the clinics, because you learn a lot about how the drugs interact with the patients and how they interact with each other and the safety profiles, but also to learn chemistry and biology behind how to develop and improve drugs. But I also knew that in order to become a good scientist, I had to do a PhD, of course. So after pharmacy school, I decided to, because I'm from the countryside of Brazil, in the middle of the mountains. It's a state named Minas Gerais. And there's a very good pharmacy school, quite old, the first one in Latin America.
Wow.
But I wanted to do something different. So I went to the University of São Paulo, because there is the financial center of the country, a lot of money for science, and you cannot do science without money. So then I decided to go there for a master's and PhD. And then by chance, I found out during a winter course that I did in the University of São Paulo, a researcher, a very renowned researcher, Giulio Votarelli, that worked with cell therapy to treat autoimmune diseases. And then during that time, I could do some interviews with him, and then I did my master's and PhD in this area. But after finishing my PhD and publishing good papers showing how this therapy works, and then going back home and talking to my family, they're like, okay, so when is this coming to everyone and being used as a drug? Then he said, it's not coming because it's just an academic development. We are learning how this therapy works, and to make it safer, of course, and to improve it and to make it more applicable to more patients, but it's not going to be available for everyone. And then I realized that, okay, now it's time to go for a change and do something that is more applicable, industry oriented, that we can create a drug and apply that to more people. And then I came to Sweden, where I live now for 7 years, to do a postdoc. And then I decided to join here because it was a drug development project. So it was two positive things that came at the same time. My supervisor was a very busy guy working the hospital side at Karolinska University Hospital, so he couldn't supervise his PhD students as much. Then I had this experience of supervisorship very early on in my postdoc career. And also because my project was to develop a bispecific T-cell engager. So it was basically to design a new drug. Of course, it's very preclinical. It doesn't mean that it's going to be used, but at least it
kind of idealistic.
Yeah, exactly. So it's something that at least I could use all the knowledge that I got from the pharmacy school and also from my PhD in immunology to apply all of these all together. So that's making a long story very short.
No, I love the long stories. So how did you find out about the opportunity in Sweden?
Yeah, I mean, I think that a lot of things in my career, it happened by chance, I would say. For example, this winter course that I did, it was just because my cousin was going there, and then she told me about it, and I applied, and then I met my supervisor to be in a very informal way, and then things happened. But it happened because during my PhD, I went to France for 1 year.
Okay. Yes, I saw.
Yeah, so it's in Brazil we have a lot of agreements, bilateral agreements. So a lot of students coming to the University of São Paulo and us going abroad. So some friends came, and then I went to France, and then I really enjoyed the environment and the facilities of doing research in that setting. And then, for my postdoc, I want to go abroad. I applied for several positions in the US, Germany, basically everywhere that I could find good supervisors that had money to do high-level research. And then it happened that in Sweden, it was a good, let's say, match of
Yeah.
being able to supervise the students, which is something that I would love to do early on in my career, like I did, and also to work with drug development. So it was just by chance. I applied to several labs. Most of them said, no, we are quite full. Or, oh, yes, you can come, but you need to find your own funding. And then
Yeah.
it's like, no, I don't want to do that. So then
Yeah.
at the end, I ended up here in Karolinska Institutet in Stockholm.
Sounds like you found your home though.
Yeah, I
Like
mean,
you said, these things happen by chance, happen for a reason.
yeah, the plan was to come here, do a 2 years of training, and go back to Brazil. But then I started applying for grants and like working in different projects because I was supervising some of the students, not supervising in the paper, but like co-supervising. And things that started to work very, very well, getting some grants, publishing some good papers. And then here I am 7 years later. And now I became Swedish citizen. So I'm
Oh,
stuck.
congratulations. You really are. It really is home now.
Yeah, exactly.
How important, there's a few things that you've mentioned there about your superiors and supervisors and how much you enjoy supervising. How important is that mentoring element to people when they're early in their career?
It really depends of what you want to do with your career and what is your profile. Let's say like that. Some people are natural like leaders, and they like to be, and they have a good feeling about it. But others, of course, they require more training. But in my case, I always wanted to supervise, and during my PhD, I wanted to be a professor and lead my own team, and train people and teach and do all those things. So I started to do that as early as I could. But some people they don't like that. So it really depends on your career. If you need to find where you feel more comfortable and what you like to do. Some people they like to work in a more organized way. For example, in our CDMO, you have all the GMP people working with the production, quality assurance, and control. That requires a specific skill set that you don't find in everyone. To be very organized and planned and follow these structures is quite structured work. But of course, for some people, that's something that they will never do.
Maybe too rigid for some people.
It's too rigid, and they don't like to be in this kind of monotonous work. Some people would say, because you follow these strict guidelines and have to report every single deviation and a lot of paperwork and planning. Which is good. I mean, I also like that. But of course, I see myself better in other positions. But happily, I am right now. But that's one important thing. Like is to find something that you like to do and where you see yourself. Maybe some people they like to do research and development and be a lab technician. Some of them they like to grow in the career and do other things. It's really a matter of trying to match, and that's the most complicated thing I see in biotech is to match expectations, qualifications, and your role. It's complicated because if you hire someone with a PhD level to run a GMP facility production, some people maybe would like it, but I would say that like 90 to 95% they would quit in the first 6 months because they did a PhD, they were expecting other things, a more dynamic work, and then
more on the discovery side and the innovation.
Exactly. So then that's not a good match. So that's the complicated thing with careers. It's to understand what you want and match your ambitions and expectations with the market, with everything else. That's the complicated part.
We talk a lot with the clients that we work with in search projects about technical skills are important. The cultural fit is more important. Getting that it will be, that's the reason that people tend to work or not work long term within a business.
Yeah, I totally agree.
Yeah. So have there been, what would you say are the key learns that you've had in your career today?
I think adaptability is the key one because I didn't say, but at the end of my master's, my supervisor unfortunately passed away. So
Oh,
a lot of changes happened in the department, in my personal life, because it's a big shock. I didn't know if I should change department, change things, but I know we had this agreement with France that I could go there, and then
yeah.
I also had personal problems because my dad had prostate cancer and it was not doing so well for him. And then, I was in the middle of all this storm. And then you
Yeah.
just need to adapt personally and professionally to what it comes. And that was quite interesting as well because during this time in France, it was a quite new technique at that time, like 10 years ago, to do a T cell
Yeah.
next generation sequencing. So we
Yeah.
doing this massive parallel sequencing of thousands of T cell receptors, and then we decided to do that for the very first time, and we
Mm.
didn't know how to do it. We didn't know how to analyze it. We didn't do anything, but then we just adapt to the needs and to the cool thing that could be, and then we just made it happen. And then you just
Yeah.
have to adapt to what is coming to you, and that's something that is very important for anyone's career, I would say, is just to
Yeah.
adapt to what comes to you. And because especially in science, there's a lot of new things coming all time, new techniques, new ways of using this technique into other formats. Now we have AI that came
Yeah.
to completely shape the field, and you just need to adapt yourself and learn new things and keep yourself updated. That's the key thing for any career.
And be prepared to take risks. I feel like in your story, there's a lot of times when you had to be brave to take the decision that you did.
Yeah, yeah.
um, when it comes to taking risks as a scientist, due diligence, looking at the facts, weighing it up, do you do that formal risk mitigation, or does it come from emotion? Does it come from a feeling?
I think for me personally, it's more like a feeling. It's
Yeah.
like, yeah, as long as you are motivated to do something and you are willing to work hard to achieve your goals, it doesn't matter what risk you take. Um,
Yeah.
I think I have taken quite a lot. Before joining Curacell, I was working in another biotech, which was my first work outside academia. The name is Anoka. It's a TCR-based company that is also here in Stockholm area. So during my postdoc studies, I had different collaborations to test this new drug.
Okay.
And then I was testing this drug with another postdoc from another group. And then he told me about this company that they had this position. Lucas, you wanted to try something new. Why don't you apply? Then I did it. And then I got the position. And then I worked there for 3 years. So it was already taking a risk because I was really happy
Yeah.
in my work. I mean, I was really happy in my postdoc. I just had a
Yeah.
grant approved, quite good money, good papers coming out, a lot of good things happening. But then this opportunity to join industry in a good position, because the position was to lead a team there. That
A big
came,
step
and
for that first role in industry.
yeah, exactly. So
Yeah.
that came quite early, I would say, in my career, and then I decided
Yeah.
to take that risk, like changing a place that I was very
Yeah.
happy and successful, to try something new, completely uncharted waters, let's say.
Yeah.
um, but it just ended up very well because, of course, if you work with good people, you work hard, you will always find a way. And then I wasn't happy there as well. And then I was approached by some headhunters like, hey, we have this position as CSO at Curacell. Would you like to change? I was like, yeah, I mean, I'm very happy here doing what I'm doing, but why not to take a risk and try it out? Because I mean, if it
Yeah.
doesn't go well, you can always go back and find
Yeah.
and find something new, basically. So taking risk is good if you are willing to work hard for it and understand that, yeah, if things go bad, you can just change. And there is nothing in life that you cannot learn. You can always learn new things.
Yeah.
You can always adapt to new things, and the more you understand that, the easier it is for you to grow.
Yeah. Yeah. And that's something, there's always that appetite for learning within our industry. Things move so quickly. Things are
Yeah,
always changing.
things
It's
are
definitely a
changing. Yeah,
quality that's
exactly.
instilled, isn't it?
Hmm.
I'm interested. Something that I'm often hearing is that switch, whether people are looking at academic careers or whether they're already in academia, making that switch into industry. I sometimes hear people talking about the fear of the commercial element of industry putting them off. How did that affect your decision making to move into industry?
Yeah, that's a good point. I mean, like I said before, I always wanted to do academia and to be a
Yeah,
researcher, become a professor, because of the freedom, because of all these opportunities to try new things and do something and apply for a grant to, to have a groundbreaking research or have like this kind of high risk, high reward experiments or projects. But also at the same time, industry gives you more organization, more money to try new things, and that's
yeah,
something that people sometimes in academia are trying to do because it's quite important for people that are trying to change from academia into industry. Is that you need to have the package of skills that is
mm,
required for that, and it's quite sad sometimes to hear that people get disappointed that they hold a PhD and then they apply for some positions that they are overqualified.
yeah,
So, for example, it's a position that it only requires a master level, and then
yeah,
people they apply, they get a quick rejection, and they get very frustrated. But it's not because they don't have the skill set. It's just because it's not a good match for the
yeah,
position. And it's
yeah,
very important for industry to try to make this good match. Otherwise, you waste a lot of time and money hiring
yeah.
people that will not be a good fit. It's like hiring this PhD level, super fancy lab technician to run GMP production.
Yes.
It doesn't
Yeah,
make much sense. And
yeah,
that's something that people trying to change from academia into industry needs to have in mind. You can always
yeah,
find a spot. There is a lot of opportunities, but you need to offer the skill package that is required. And of course, there are some hot topics nowadays, like cancer immunotherapy,
yeah,
some specific areas of biotech. It's really growing quite fast and with several opportunities. But you need to offer the skill set that is necessary, either by knowing techniques or by knowing how to do a specific analysis. But this leap from academia into industry for sure can be a bit complicated,
yeah,
depending
yeah,
on expectations of the personnel.
yeah. And we maybe need to do some work in industry to make sure those expectations and qualifications are matching, give the right
Yeah,
opportunity.
and it's quite interesting that at least here at Karolinska Institute, they have some internships for PhDs and postdocs to go
Yeah,
and try out industry for a couple of months. So
yeah,
that's an agreement between any biotech or industry in Stockholm area and Karolinska Institute. We ourselves at Curacell, we have had a couple of interns, which is very good for you to see if you're going to like the work,
yeah,
if you're gonna like
yeah,
the environment, because it's not an academic group that you go, you publish a paper and you publish a poster and you do things. No, it's another different way of documentation of organization, and then some people love it, like myself,
yeah, yeah,
but some people they don't like it at all. So
yeah,
it's yeah, that's a good thing that you can go and do an internship, or for example, if you are interested in doing something in industry, you can ask for a leave of absence or apply for those internships and
yeah,
just try it out and see if it's a good match.
yeah. I am seeing more and more universities linking with industry to create those opportunities. Sometimes in the UK, they even do up to a year's internship with an organisation to, yeah, really get under the skin of what it is to work in industry.
Yeah, but and I also think that industrial PhDs, it's something that it might grow in the future,
Uh,
because
Yeah,
we need to align all this freedom from academia and this free thinkers to bring
yeah,
new ideas into industry. So it's something that I think it's going to grow quite
yeah,
fast.
yeah, absolutely. So if you were to think back to Lucas back when he was at university, or to those scientists who are in their early career, what one piece of advice do you give to yourself or those early in their career?
Oh my God, I have no idea. Yeah, I don't know. I think everything was, it worked so well that I think everything was quite smooth. And but I think like learning new things and also yeah, trying to be updated to what's
Yeah.
going on. That's something that I see nowadays, even unfortunately in PhD students, that they are not updated to the most current techniques.
Really? Wow. Okay.
Yeah, to the most
Yeah.
current science and the papers, because of course that requires a lot of effort because it's a lot
Yeah.
of
It
papers being
moves.
published. It's
And it
insane.
moves insane. Yeah.
It moves so fast, and the amount of data that people can generate right now with all these omics platforms, transcriptomics, genomics, spatial biology,
Yeah.
all those things, they move really fast, and people, they don't get updated on what's going
Yeah.
on. And because things move really fast, it's quite fast for you to be behind.
Yes.
So that's
Yeah.
something that I would have thought myself, is like pay attention to this,
Yeah.
because you need to keep yourself updated all the time. Otherwise, it's a shame and a waste of time. I recently finished reading the book of Kathleen Kakariko,
Okay.
biography. She won the Nobel Prize a couple of years ago with the messenger RNA vaccine that was
Right.
used
Okay.
for the COVID pandemic. And one thing that she wrote in the book that I found quite interesting, that she was so disappointed that some professors and famous investigators, that after they achieved like a quite high level in their career development, they just stopped to update themselves, and they would just say
Oh,
things
really?
that were completely outdated. And she's like, yeah, how is this possible that how people in this area can be so
Yeah.
outdated? Because you just can't. You just
No.
can't.
Got to keep current.
Otherwise,
Finger on the pulse.
yeah, exactly. You need to be in the forefront all the time if you want to create new things and to be
Yeah.
valuable, basically.
Do you have any predictions of what the next big thing will be? Where do you see the most scientific changes happening in the next few years?
I really like spatial medicine, and I think it's something that it came to stay with all the spatial
Yeah.
transcriptomics and proteomics, and I think that clinical application is really close to happen. And yeah, I think we are going to see more and more. But of course, AI, I'm a big fan of AI. Some people don't like it.
Yeah.
Because I mean, recently we saw these Nobel Prizes that are very linked to artificial intelligence, and it it
Absolutely.
it also came to stay. But of course, spatial medicine relies 100% on artificial intelligence, and
Yeah.
I think that's one of these groundbreaking technologies that came to
If it can speed
change
up the
everything.
process, make it cheaper, make it more accurate, that can only be a good thing for patients, can't it?
Yeah, exactly. Yeah.
Okay, so what's next? What's next for Curacell? What's next for Lucas?
Yeah. So for Curacell, we are in a very interesting phase of our development. We are completing now all the paperwork for clinical trial application. So as I said before, we have a quite unique expansion protocol to go to infiltrating lymphocytes. And we saw in our patient name treatment some good indications that this could work very well for cold tumors. So now we are finishing the paperwork, and hopefully by Q1 2025, we are treating the first prostate cancer
Oh,
patient.
so exciting.
Yeah, that's going to be quite interesting.
And it's
And
just around the corner.
yeah, it's just around the corner. Yeah, we just waiting for the final papers to be fixed, and have everything in place, and just applying for the clinical trial application. And that's going to be our next phase, is to see if our development is going to lead to some improvements in the people's lives.
Yeah. Very exciting.
Yeah.
And this will be your second clinical trial.
No, this is our first in human.
First in human.
Yeah, because the patient named treatments doesn't count as a clinical trial.
Right. Okay. Yeah.
Yeah. So that's not the same. But it was good to have used our product before to show that it's safe. So among all patients treated, we treated 11 patients, different indications. all of them with cold tumors, all of them, unfortunately, really close to death. It was super safe. We treated to up to 9 infusions because that's our clinical protocol. It's based on like several infusion of TILs. Because our approved T-cell therapies, they rely on one single infusion. That works really well for blood malignancies, as I said, for CAR Ts, and Iovance for melanoma. It's one infusion. There is also a TCR3 product from Adaptimmune that is approved. There is one infusion as well, but they are all on this hot tumor area. And we think that to reach good response rates in cold tumors, we need to hit more than once with TILs.
Right.
So the idea is to give more than one infusion. Like when you have it's normally you take more than one pill, right? To take several pills. So that's our thinking that with TILs, we need to optimize the expansion protocol in that sense, that we can generate good products in fast time, that it can be delivered more than once to the patient. So that's our next phase.
Very exciting. Can't wait to hear the news that's going ahead in Q1.
Yeah.
And how will you continue your learning? We talked about that appetite for learning. What's next for you?
For me, is to bring Curacell further. Now we are focusing our development just in the expansion protocols, but because of the continuous changing the field, there's a lot of new things coming up. Just came from the US last week, where we had the TIL Summit. So all the key players and leaders in the industry, they were there, and then we could see what's going on. And everyone now is doing different engineering of TILs. They are doing selections of different subtypes, trying to make TILs safer. That's always the first goal, to make safer for the patients, and also to increase the efficacy. So that's our next step for me and for Curacell as well, to develop further, to engineer the TILs and do some selection and improve our expansion protocols to make it safer and more efficacious.
Yeah, fantastic. Well, thank you very much for joining us. It's been a pleasure.
Thanks for having me. Yeah, that was really nice. Thanks a lot.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.